CBD is well studied, certainly by the oral route, and less so by the inhalation and dermal routes. And it does have quite an interesting toxicological profile, which is one of the main reasons we decided to talk about it today. Its pharmacological action has been exploited for therapeutic benefit, and I'm sure we'll hear more about this during the summit. But for example, as an API for treatment-resistant and severe childhood epilepsy. And there's just a note here for toxicologists out there: we have to be very careful when we're carrying out literature searches for CBD, because there are a number of potentially useful CAS registry numbers for CBD and its isolars. So you have to make sure we search on all of those to capture all of the potentially relevant information. And when we do find some data, we have to be carefully consider what was the actual tested material in those studies. So for example, sometimes it might be on a mixture, like Sativex. But unfortunately, a lot of the earlier published data studies, they lack detail in what was actually tested, the purity of the CBD and the impurities involved, and that could have a big impact on the study findings. But we have found to date that the purity is typically greater than 99% in the more recent studies, and the only notable impurity that's identified is THC, and that's typically less than 0.5%. So the next few slides are going to be looking at the major toxicity from the dermal, inhalation and oral routes. So pharmaceutical companies are actually developing a synthetic transdermal CBD gel. And this pharmaceutical company has actually conducted several human studies, generally involving only small numbers of patients, and the studies of varying duration, but some are over 2 years long, and they employ a typical starting or maintenance dose of 50 milligrams per day, but the dose of CBD can go up to about 250 mg per day. These treatments are all described as being well tolerated, with an absence of adverse effects, so that's reassuring from the dermal route. One thing to consider is the dermal bioavailability of CBD is expected to be very low, as in CBD is not expected to penetrate the skin. The reason that it is efficacious in these studies is that the gel involves permeation enhancers within the formulation, and that helps the CBD to move across the skin barrier. So turning now to inhalation toxicity. Unfortunately, there's no good quality laboratory animal or human inhalation studies that are designed to assess the potential local toxicity of CBD on the respiratory tract or on systemic inhalation toxicity. In one limited human study, no overt signs of respiratory tract irritation or adverse systemic effects were seen in a randomized, double-blind, placebo-controlled pilot study, which involved the ad hoc use by smokers of an inhaler delivering 0.4 milligrams CBD doses repeatedly over the course of a week at a CBD concentration of 5% in ethanol solution. So that, although reassuring, it isn't particularly robust data when we're trying to have a good understanding of the potential inhalation toxicity of CBD. So from there, the risk assessor has to think how they can go about assessing CBD's inhalation tox. And one way of predicting it is the use of data involving other exposure routes. And this has to be done carefully, and there is certainly much uncertainty. One of the main things to consider is the bioavailability differences in humans between the different routes. So for the example, oral CBD is estimated to be bioavailable only about 6%, and that's due mainly to the low and erratic oral absorption and the significant first-pass liver metabolism. Compared to anecdotal evidence of greater than 65% bioavailability following inhalation exposure. So you can see there's quite a difference there in the absorption and the metabolism and things. So that's certainly something to consider when you are carrying out cross-route extrapolation. Okay, so now we turn to the local toxicity and sensitization data. And this local irritation doesn't seem to be a particular issue with CBD, and that's of the respiratory tract from the inhalation study that we talked about on the previous slide, and also the available skin and eye irritation studies. For example, there was no evidence of skin irritation in a number of studies in humans involving acute or repeated dose topical treatment with a gel supplying CBD at up to 2.5%. There was also no overt irritation in rats receiving repeated skin application at concentrations up to 10%. Unfortunately, we haven't found any data on the skin respiratory tract sensitization or oral allergy. So that area is still a bit of an unknown, unfortunately. Okay, turning now to oral systemic toxicity. As mentioned before, there are a large number of repeated dose oral studies in human volunteers and specific patient groups, which is great news for the toxicologist. Most of these found an absence of significant adverse or side effects in the treated subjects, and that was when they compared to the placebo controls or to baseline evaluations. And the effects seen were generally described as mild to moderate and transient. Indeed, published reviews, including those by the World Health Organization's Expert Committee on Drug Dependence, describe oral CBD treatment at therapeutic doses as well tolerated and with a good safety profile. So again, that's certainly some reassuring information there. And in some of the more recent studies, 20 mg per kg body weight per day, it's about a gram a day for a 50 kilogram adult, has been approved by an independent data safety monitoring committee, which is a requirement of all clinical trials, as a safe dose without unacceptable side effects. So that's quite an important dose to remember, certainly for clinical trials. And some of the side effects that have been seen in the studies associated with this dose, sedation and fatigue, reduced appetite, diarrhea, effects on clinical chemistry, for example, increased liver enzyme activity. But this could be a result of drug-drug interactions between the CBD and the patient's existing medications. So, for example, their epilepsy drugs. And we've also seen some blood changes as well, so reduced hemoglobin, for example. Of course, it has to be borne in mind that there is a perceived benefit to the patient of the therapeutic doses producing these, albeit mild and reversible side effects, which would certainly be seen as undesirable in a consumer. So, I mean, a cosmetic consumer or an e-liquid vapor, for example, or someone consuming CBD in food or in drops. So this means, or this indicates, that the true human oral NOEL is actually less than 20 milligrams per kilogram body weight per day. Indeed, the UK Committee on Toxicology have recently assessed CBD for use in consumer products. And following this advice, the UK Food Standards Agency recently recommended that exposure to CBD should not exceed 70 milligrams a day, which is about 1 mg per kg body weight per day. And this is related to oral products, unless under medical supervision. So I'm quickly going to talk next few slides about some of the critical oral laboratory animal data that's available. And there is quite a few studies, but one of the pivotal studies was a 90-day study in monkeys involving oral exposure to CBD at 30, 100 and 300 mg per kg body weight per day. And there was some increased organ weight changes seen, and that included the liver, the kidney and the heart. There were also some dose-related decreases in testicular size, inhibition of spermatogenesis, and some accompanying histopathological findings in the testes. So that's certainly something to take note of. There were no other treatment-related histopathological findings. And the study lowest observed adverse effect level was described as 30 mg per kg body weight per day, which was the lowest tested dose. So essentially there wasn't a dose in that study that was out without adverse effects. So now we turn to reproductive toxicity, and you would have seen from the the previous study in monkeys that effects on the testes were seen. So that's certainly a bit of a flag when we turn to the actual reproductive toxicity studies. And in some laboratory animals, adverse effects on male sexual function and fertility have been seen in oral and injection studies. And there's a possible role for hormonal disruption in some of those. However, no adverse effects on fertility are seen in rats orally administered CBD at up to about 250 mg per kg body weight per day. And that was a study by Greenwich Biosciences in 2018. So there's a bit of conflicting information out there. But also some effects have been seen on offspring development, including small testes in the adult offspring, and also embryo fetal mortality. And these have been seen in what appear to be reliable developmental toxicity studies, and they are seen at doses that are not causing significant maternal toxicity. So that means that the effects being seen are slightly more concerning. But the doses used are nonetheless higher than that would be recommended for human treatments. But what is reassuring is that CBD is not teratogenic, which means that no malformations have been seen in the developing offspring. Concerns for reproductive toxicity have also been noted by expert groups, but they do acknowledge the limitations in the data and the absence of human studies. So it is worth considering the possibility that in the future, a health precautionary regulator may suggest that CBD should be classified under the European Union CLP criteria as a suspected human reproductive toxicant. That's certainly something to bear in mind. It's a potential risk for companies. And there also is a possible link with an effect on or via lactation, and CBD is likely to transfer to the breast milk. So companies may want to consider product warnings, for pregnant and breastfeeding women on their products. Now we turn to genotoxicity and carcinogenicity, two critical endpoints of the toxicity profile of any compound. And the genotoxicity findings for CBD are somewhat conflicting. A fairly recent publication by academics from 2 European universities reports the results of their positive in vitro micronucleus and comet assays, and combined with the findings of earlier in vivo studies, indicate that CBD is a potent mutagen, and that's quite a worrying conclusion. However, Greenwich Biosciences have noted the existence of other data that suggest a much cleaner genotoxicity profile for CBD, and that includes data on their FDA-approved drug, Epidiolex. And that was from in vivo studies in rats, and also a bacterial mucosity assay, an Ames assay. And of course, this data must have been submitted to the regulator, so for example the US FDA, and therefore approved. So there is some reassurance that the regulators are not too concerned, certainly at the levels and for the therapeutic benefits, etc. More recently, the UK Committee on Toxicology has concluded that there were no indications that pharmaceutical grade CBD was genotoxic, based on the summary genotoxicity data from an adequate range of tests performed. And in addition, there's no evidence of carcinogenicity seen in rats at up to doses of about 50 mg per kg body weight per day, which is quite low, for 2 years in the diet. And on the back of that, the European Medicines Agency, the EMA, their assessors have noted that the carcinogenic potential of CBD has been adequately evaluated to be negative, which is quite reassuring. But it would be good to have a second cancer study on a different species, so for example in mice, to give us a bit more reassurance. Okay, so turning to some of the conclusions, perhaps surprisingly, CBD has quite an interesting toxicity profile. I perhaps didn't expect that as I first started to review it a few years ago. Of particular note, there are only limited inhalation and limited dermal data out there. So when you're considering the systemic toxicity of CBD, you could well have to rely on the oral route of exposure, where there's far more data available. But also you have to consider the uncertainties involved with such a route-to-route extrapolation. But certainly adverse effects have been seen in repeated dose oral studies in patients, in laboratory animals. These are generally described as mild to moderate and reversible, which is encouraging. Some of the effects, like fatigue and increase in the liver enzymes, possibly as a result of drug-drug interactions and blood effects. Although these may be tolerable for the patient population, so if they're helping them reducing the number of seizures in epilepsy, that may be deemed as acceptable. Of course, that might not be the case for the general consumer. So we have to be careful when we're using information from pharmaceutical studies and from pharmaceutical expert groups, etc., and when we're using that information to extrapolate to the general consumer. There is evidence of reproductive and developmental toxicity potential for CBD, for example, on the male fertility. And so you may want to consider warnings on your packaging to help mitigate any risk, for example, for pregnant and breastfeeding women. The genotoxicity findings are conflicting, but recently the UK COT indicated there was not a concern for the pharmaceutical grade CBD, and on those more recent studies, and there's no evidence of carcinogenic activity in rats. Ultimately, it's important for companies to carry out product-specific human health risk assessments to ensure the products they place on the market are safe for their patients and for their consumers. And the use of high purity pharmaceutical grade CBD is a very important step towards that. soon.